Okay, let's start in the afternoon with a little bit of theory about isobaric labeling. So you already learned a lot about label-free quantification from Carlo. And just to understand it again, right, we have like two samples prepared independently. Then they're also measured without any labeling, just two parallel runs, and then quantified against each other. This is, it's very easy from the experimental side, but on the other hand, it is quite challenging on the algorithmic side. And you learn things about matching between runs, retention time alignments, and all this normalization business to do a proper analysis of label-free data. But there's also MS1 labeling, so-called isotopic labeling. And the idea is that there is a label, a tag somehow introduced in the sample, in the peptides, and it's coded using neutron coding, neutron-based labeling. That has the big advantage that if you have like 1 neutron more in your atom, right, everything is a little bit heavier, but the chemical properties stay the same. And then you can do, I need my pointing hand. So then you can mix 2 or 3 of these labeled samples together. So then you have a light, a heavy channel, or even a channel in between. And on the MS1 level, they are like clearly separated, right? They're like the MS1 features, they have a distance, a distinct distance, and then they can be quantified against each other, which is a very nice way to quantify. And it's actually much easier on the algorithmic side because you just take the ratios in these from one raw file. But now we're talking about MS2 labeling, and this is chemical labeling, and these work like the basic idea is the same, to have a neutron-coded tag, but they're actually designed that they all have the same weight, unless they are cleaved. So that means on the MS1 level, right, so they are all mixed together and measured together, so they just stack up in this MS1 peak and are not distinguishable. But then afterwards, when on the MS2 level, they are cleaved, and then they are somehow separated. Sounds like magic. It's also somehow magic. It's very cool, but let's understand it and let's see what you can do with it. So this brings actually the big advantage that you can do multiplexing. Up to 18 samples can be measured together within one run. So compared to label-free, that saves a lot of instrument time. Okay, so this is iTRAQ. iTRAQ is one of these tags, isobaric tags, developed and sold by SciEx, and that it contains of these 3 parts, right? That's the molecule. We have a amine-specific reactive group on one side, then there is a balancer group, and we have a reporter ion. And actually the mass is the same. So here we see a 4-plex, and they all have the same mass of 145.1. But there is a difference because the reporter ions, they have different masses, and these balancer groups too, and just the sum is the same. And this allows then to do this multiplexing with 4 different tags, but there is also an 8-plex. Another isobaric tag is TMT. It's the same principle. It also has this, these 3 parts. It has a mass reporter, it has a mass normalizer, and it has the reactive group, which binds actually to lysine side chains or the N-termini. And here the reporter ions lie in a different range. So it's actually very similar, but you have more variations, and you can go up to the 18-plex. So let's have a look at not at the 18-plex, but at the 16-plex TMT. Again, right, reporter normalizer reactive group, and if that reacts with a peptide, then we get a TMT labeled peptide over here. So this is then attached to all these peptides. And in the MS2 fragmentation, this reporter is cleaved and measured and ends up in the reporter channel. So this is a 16-plex. But when you count, how many atoms does it have? Well, you don't have to count it, but it's for sure less than 16, right? So there's another trick there, right? So it's not that there's only like one neutron more and more and more and more, but there's also a difference between actually, is it the nitrogen or is it the carbon which has one neutron more? And there is a slight difference in the mass. To be precise, 6 millidaltons, and this is actually then allowing us to have a 16-plex with only 9 atoms. Very cool. I like it. So that's how it works. That's how like the typical experiment looks like. You do your sample prep, and like always, there is one red sample and there is a blue sample, and they get labeled. One with a tag that has a light reporter, and the other one has a heavy reporter. Again, right, the total mass is the same, and we mix it together and get this very nice purple mix. So on the MS1 level, it's still purple, right? Like we can't distinguish it. But after MS2, after the MS2 cleavage event, then in the reporter region, right, which was like 120, 230, something like this, depending on the tag, then you can see there is the blue and the red tags, and you can quantify by just taking the ratio. So sounds perfect, right? Very nice, and also actually very popular. But there is some things that you have to consider when you want to do a TMT labeling or iTRAQ labeling. So of course, there's the sample prep side where the labeling efficiency is important. Then there is actually isotope impurity. So these tags have impurities. There is this famous ratio compression, which I was already talking about yesterday a bit. There is also, like, depending on the intensity of the reporter channels, there can occur some problems. So I will talk about all that now, and then we will also see how actually you can do comparison between labeled or multiplexed samples. Let's have a look at the isotope impurity. So I made this 4-channel representation. We have 4 channels, the reporter channels, and of course they have colors like always. But what can actually happen is that in the channel 1 there is some contamination, right? Some of the tags from channel 2 end up here, and the other way around. And this can also happen in the other channels. Reasons for this is it can just naturally happen, right? That there is some like isotopes evolving. There is also impurities in the labels, and this can cause these impurities in your channel. This like contaminations, and this can actually sum up to like 10 to 20%. But the good thing about it, if you know it, then you can handle it. And actually, since it's batch dependent, so on your batch, you usually get such a table. It shows you, gives you some correction factors that have to be applied, and it just tells you, aha, for this reporter, this iTRAQ reporter 114, there is these corrections which you have to consider to get the perfect quantification results. And Daniela will show you how to put these numbers into your MaxQuant configuration. And then you can be happy and you can look at the next problem, which is called the ratio compression. Okay, so ratio compression is actually caused by the following phenomenon. There is, of course, co-eluting peptides, which have actually overlapping isotope patterns, or very close isotope patterns. And if a precursor is selected for fragmentation, there is the window, the quadrupole. Usually you take like 1.4 Daltons window size. But I mean, the quadrupole is not perfect, right? It's not like it looks here, right? It's like there's a hard cut. So it's anyway a bit so you get some other peptides in your window, which can be actually very useful in some cases, because MaxQuant, you can actually take them later and do second peptides and get more identifications. But in the case of isobaric labels, that's not what you want. Because you only have these reporter channels, and probably they also end up in this reporter channels together with the selected peptide. So to understand it a little bit better, I want to show you this experiment. There is a mixing experiment. So there's some yeast. Yeast is black. And that was mixed in 10 to 4 to 1 ratios, and the other way around. And there is also a human cell line, which was mixed in the first 3 channels on top of that. So if you now would, in an ideal world, if you would just do the analysis and quantify the ratios between the yeast peptides, then you should actually exactly get out what you put in. And the ratio in such a case is this complicated formula that you divide just I call it signal of one channel by the signal in the other channel. But we have co-isolation. So what happens is that also the yeast peptides, which were co-fragmented, end up in our channels, and they end up here. So they push everything up, right? So we get, and this is 1 to 1 to 1. So there is no difference, but everything is pushed up. And if you like do a little bit of math, then you can see that actually this ratio will be smaller than the ratio in the ideal world. So usually it's compressed. It also depends, right? Like what is the contamination? Is it like really 1 to 1 to 1 to 1? It can be also like the ratio can also be like bigger sometimes or whatever, but like in average it's compressed. And this can be a problem, and there is some attempts to solve that. So one idea is to do the MS3 spectra. So MS3 spectra are actually taking MS2 fragments and fragment them again. So usually you can do like different collision energies first, or different fragmentation techniques. So for example, in this case, MultiNotch, they do first CID fragmentation and then from these MS2 spectra, the most abundant fragments are selected and refragmented, and from those are the sorry the tags end up in the channels for quantification. And this in this case, so there was also like this mixing experiment, and you can see the mixing ratio was also 10:4:1. And in the case of MS2, because of the compression, it was like 4.6:1, but with MultiNotch MS3, it was actually correct with 10:1. So this is an early approach using MS3. There's more elaborate methods now, which do like real-time detection and selecting the fragments. This MS3 technology is getting better and better, and it's also widely used. But there's also actually the easy tag I was talking before. So this is a like different tag, which does not use the reporter channels for quantification, but it is using the fragment ions. Sorry, no, on the MS1 level. So it's cleaved up, cleaved. And then you see you get this splitting on the MS1 level first, and after that it's fragmented with a higher energy, and then you get like proper MS2 spectrum. So as I said, this also needs some adjustments in the DDA acquisition strategy, but it also works. But it's, as far as I know, not yet commercially available. Good. So we now solved that problem. Let's see what else you have to consider. And there is the reporter ion saturation phenomenon, also called coalescence. So in this case, for example, let's have a look at this channel, right? This is 130 light and heavy, right? So very close together, these few millidaltons. And what actually happens is if the intensity in that channel goes up, right? So it's hard to see, but this is like magnitudes higher than that one. These two peaks, they merge together in the system, and this was a orbit trap, and you can't distinguish them anymore. So there is no ratio. These problem can be solved, right? So it was detected, and it could actually be solved by just changing the acquisition parameters. So there is this maximum injection time parameter that actually determines how many ions will you get in your reporter channel, and that was then just reduced a little bit to not get these very high intensities, and that would actually like then solve the whole problem. Okay, so sounds good. You know what are the challenges, and you know how to deal with them. But it can also be that you don't want only to compare what you actually mix together in the multiplexing, like these up to 18 samples, but you have several runs, right? So you want to compare runs of multiplexed measurements. And there is some things that have to be discussed if you want to do that. So we're now like slowly moving in the direction of what Carlo was discussing about label free, because this is now again labeling, comparing runs between each other. And as Carlo said, there was this matching between runs idea in the label free. In the case there is in one run a MS2 identification, and in the other one not. So, but the isotope patterns are there on the MS1 level, right? So illusion times and masses, they fit. And then the idea is, okay, well, there was no MS2 event, or the MS2 event was too bad. So we just transfer this identification from one side to the other. And this can, of course, also happen in our runs with the multiplex sample. So what is then done, or what was implemented for this paper, is to do the same, right? To just transfer the identification. So, for example, here in B, the upper one, this peptide was identified. It had a good Andromeda score, and this one and the other one had a bad score, and there was no identification, even though it's kind of like obvious that they are similar. And so this isobaric matching between runs then now allowed to actually identify also this one and then get the quantification in the reporter channel, which you can see here. And in this case, we see actually this was an experiment with 8 isobaric labeling batches, and the number of, it shows the number of peptides shown in N out of 8, right? So actually this part here, these bars are the most interesting ones. So how many peptides were found in all 8 raw files? And if you compare the orange bar, which means there was the isobaric matching between runs, and blue, it was off, then it's kind of explaining that this is a very good idea to do, to boost your identifications, or actually multi-quantifications. Okay, so what else? What else, Carlo? What were you talking about? You were also talking about, yes, normalization. That was a big topic in the label-free talk, and this is also a problem in the case of well, comparing isobaric labeled samples. So here we see different cell types color-coded. There's this 8 different cell types from 2 experiments. And the experiment 1 is the squares, and experiment 2 are the circles. And we see there is strong batch effects, right? So the circles are actually all hanging out here, and experiment 1 is up there, and there's no real structure, and the cell types are somehow not clustering together. So the idea here is to have a normalization for the channels, for the TMT or iTRAQ channels, by using actually doing it on the PSM level. So taking the precursor ion intensity, and this is multiplied with the fill time. So this is actually a very good measure on how strong the signal was. And the stronger the signal was, then probably the ratio is also better. And using this information for these channels, normalization factors are calculated by applying a weight median. And then after normalization, we can see that these cell types now clustered together. So the colors clustered together, and the batch effects are very good removed. So in this case, that's the same. It's the same data, just a different representation showing actually the median of the variance. And again, right, we see that we now have compact clusters and a much smaller variance within the cell types. Okay. That's all from the theory.